Hola, buenos días. Gracias por venir. Thank you for coming. And well, it's the first talk of the Delini Symposium. So people are coming. I'm just waiting a little bit, to be more people here. And I will talk about species delimitation, a little bit about species delimitation. And first of all, when we are talking about species delimitation, we need to talk about what species are. So species is the only taxonomic category that's close to reality. So the other ones are all widely recognized as artificial. And it's also the central unit for systematics and also applied in all biological sciences. The thing is that in most of cases, the limitation of species is simple, but in some cases, in special cases, it's quite problematic, especially when morphology has a limited success. When we are talking about species, we also need to talk if species are real, because if they are not real, we don't need to talk about the limitation. So are species real? Do species exist? They are just human constructions. If they are human constructions, that's fine. If you are able to identify them, that's okay. But I don't believe more in this. Actually, I believe species are real in a biological concept. So I think they do exist, and the proof that we have is just the cohesion that the genetic cohesion that populations of a species represent. So in this cohesion, it's maintained by the control of gene flow through mechanisms of isolation. So even if we know that some populations of different species may hybridize or have gene flow between populations of distinct species, we see here in this white arrows representing this interspecific gene flow, it's much less frequent than gene flow between populations of the same species. So if they try to cross only between themselves, it seems that they are a unity, a biological unity. So that's why I believe species are real. So if we think in the fact that species are real, we can talk now how to delimit them, right? And for me, it doesn't matter if you are more typologist or if you have a populationist more thinking. For most of the cases, as I saw, the delimitation will be simple. Most species are quite easy to recognize. But there are two examples when it's quite complicated. The first one are the species complexes. Thank you. On species complex, we are talking about early diverging lineages. So things that are closely related, and if they are closely related, they should be similar. So here you have an example in Trichocentrum of a student of me, Camilo Junior. We see a lot of variation, and how many species we have, that's complicated to say. And here you have the other side, cryptic species. In cryptic species, we have the opposite situation. So what we have here, you have things that are quite similar, but they are not necessarily closely related, as in this example in Nervilia by Gale. So these plants look similar, they had a single name, but they are not the same thing, and they are not related. They are in different places on phylogeny. So in these cases, especially, you need to do something more than just morphology. Here an example of South American species. It is Encyclia argentinensis. That's nice because you see a lot of variation. I don't know if you see that, but I see that. In color, in shapes, it's more clear here when you see the lips. Look at this and this. Look the shapes of this lateral, the terminal lobes. Anyway, that's quite distinct from each other. And it's also interesting that this name, Encyclia argentinensis, has 11 synonyms. And how can we deal with this? How many species are inside our Encyclia argentinensis? I think that we should take these names, all these names, 12 names, as hypotheses. So there are 12 hypotheses about Encyclia argentinensis plus the other hypotheses that are all the same thing. And how should we deal with the situation? For me, the only scientific way to deal with this both situations, the species complexes or cryptic species, is using different tools. So using different tools together to try to solve, to solve the taxonomic problem, trying to satisfy as many as possible of the species concepts. You know, we have a lot of species concepts, and they are quite distinct from each other. And if we do several distinct approaches, we can satisfy actually different species concepts, and that is the idea. Going to the first example, study example, we are showing here the Epidendrum latulabri complex. These species are all almost endemic to Brazil. They belong to the deforming group that's widely distributed in Neotropics. You see, they are all green, look the same, but no. We can see here the lips. Some are similar here, here, or here. Some are maybe distinct, and that's the point. How many species are here? First off, what happened? That's fine? Yes. First, we had 10 names for this, and we tried just to check morphology before. So we proceed with geometric morphometry analysis. And you can check here the variation in shape from here to here, and this axis, and here the variation in the lobe, divided or not from here. What we found, Epidendrum amajoriense and Anatopedium are overlapped, and Epidendrum tiagoid, pseudiforme, and Campacci too. We also see some overlap in Epidendrum latulabri and Pessoa. That's interesting. So first of all, probably it's the same thing, and probably it's the same thing, probably it's the same thing. This example, the last one, I'll show you some detail. This is Epidendrum latulabri in ABC. This is Epidendrum Pessoa and GEF. They look similar, no? But these ones are smaller, just a little bit smaller. And there are some, this is from northeastern Brazil, this is from southeastern Brazil, more or less, or southern Brazil too. We now would like to check if these groups that overlap form monophyletic groups. So we proceed with phylogenetic analysis using all these markers here. And then we see that Campacci, tiagoid, and pseudiforme form a monophyletic group here. And also amajoriense and Anatopedium, so confirming what we found here. But for Pessoa and latulabri, we don't. So the morphology, the similar morphology was just due to convergence or parallelism. But maybe no. Let's check. So we proceed also some cytogenetic data, just to check also not only chromosome numbers, but the distribution of GC rich regions in the genome. Like, it's just like morphology of morphology of the genome, how the genome is organized. So we proceed with CMA DAPI banding. Here I'm showing just CMA bands, chromosome, that show the GC rich regions. And we found interesting, it's just not those five individuals, there's several individuals. All individuals that we analyze of Campacci, pseudiforme, and tiagoid have the same pattern of distribution in CMA DAPI, 2 terminal bands and 14 pericentromeric bands. I forgot to say that all specimens analyzed have the same chromosome numbers. And then we found the same to amajoriense and Anatopedium, just 10 pericentromeric bands. That's fine. It seems that we are pretty sure that they are the same thing. They are able to cross. They have similar morphology on genome. And then we also found that Pessoa and latulabri doesn't match. So they are not probably they can't breed. They can't breed probably. Yeah, despite of this, have the same chromosome number, they probably can't breed. It's too distinct, the pattern of CMA bands. It's published in this manuscript. We reduce it to 7 species. Here we have 4 species that are from Atlantic forest, 5 species from Atlantic forest, and 2 species from Amazon. And I will talk about how this new circumscription can change something in conservation. We have another publication, I will show you then, on conservation about Epidendrum species from Brazil. And we use some ecological niche modeling too in this publication. And for Epidendrum deforme sensu latem, the new circumscription, it is considered least concern. But if we use the old circumscription, they will be all endangered. but it's just artifacts, And the same thing about conservation. If you think on 2050, this will be the nice places to conserve the species for conservation of this species. But if you check the old circumscription, look at this. I don't think it's a good place for conservation of this species. So, how try to organize better the circumscription of these species are important also to conservation of this one. We also proceed some biogeographic analysis with this group. And here we have this dating analysis. We see that the Amazonian species are not sister to each other. It's very nice because we expect they will be sister to each other, the Amazonian ones. One are here, Amapenzin, the other is Atausani. And they split from the sister one, the relative, the Atlantic forest relative, very recently, like 1,000 million years ago or less. That's much more interesting because we know the past connections about Amazon and Atlantic forest. So we also proceed some analysis on ecological niche modeling for the past, and we were able to see how it works. For more details, I have a lot of things to do, to say today. So there is a publication for that. This publication is here. We are talking about how this old distributions may have been important for diversification of this group. And here the paper on conservation that I just told you. The next steps on this group is to use another branch of species concepts more linked to ecology, using more niche as a way to separate species. We will expect, based on theory, ecological theory, that closely related species will tend to have distinct niche. Why? Because if they have the same niche, there will be more, there are more possibilities of gene flow. So they are avoiding gene flow. So they will tend to have different niches. And you have very nice results. Actually, for most cases, the overlap of niche is very low, except for this. But if you check the opposite, that's pseudoforme with Pessoa. If you check pseudoforme with Pessoa, in the opposite, it's low. So it's because one is inside the other. So I don't know if it's clear, but well, that's quite interesting that for most of the cases, there are no niche overlap. So we are working on that, maybe a publication soon. Migrating to the other side, cryptic species, that's another nice example. I have Epidendrum fulgens, a very common species in the coast of southeastern and southern Brazil. It's a orangish flower, yellowish flower. From this region, it's widely used as cultivar for gardens. And then in 2011, we found it. This grows really close to the beach, like in the sandy, white sandy, in the beach, like. And this other population was growing rocks inside the country, very distinct environment, dry environment. So is it an disjunct population of fulgens? We try to investigate it using also an integrate approach. First, we use linear morphometrics, and you found that the thing we have here in the circles were more similar to Bonicio luteum, and one with two, CV2 and CV3 show the new putative new thing. These circles are more similar to denticulatum. This is denticulatum, and this is pseudoluteum. So based on morphology, it's not fulgens. It was the first result. So now we checked about monophyly. And then we have this three, and it's also not close to fulgens. So it's close to what we found in morphology. So it's more closely related to denticulatum and Bonicio luteum too. So then we were not like totally agreeing with what we're seeing because they look the same. No, something is wrong. We are doing something wrong. So we also produce a population genetics a study, and we saw that there's no gene flow among this population, any of the other. So it's very isolated too. So then we decide to describe it. This is the publication. It really looks fulgens, looks like the same thing, but it's not. It's also interesting to say that if we include Epidendrum fulgens inside Epidendrum flammum and Epidendrum fulgens, it to be considered least concern. But as a distinct species, it was considered as endangered. So it's, again, how a distinct circumscription could reach different conservation status. Here, I just highlight some this distinct morphology that we talked in this original paper here. That we talked was just variation, but now we know this is actually a hybrid. It's actually, thank you, it's actually a hybrid, not exactly a hybrid, but introgressed individuals of Epidendrum flammum with Epidendrum secundum. And I also would like to talk a little bit about this species, that weird species, widespread, that maybe Eric would split it, but it's very weird species because it like to breed with everything. Just in Brazil, you have 5 hybrid zones with distinct species, including Epidendrum flammum. Every time it reaches a place with another species of this group, it breeds. Right? So we have some results that indicate that actually Epidendrum secundum is a paleohybrid. It's distinct to a neohybrid. It's something that have a hybrid origin, so it's a paleohybrid. The idea of this came from a friend, Felipe Nole, that's also an author of this paper. He's a cytogeneticist. And there are asymmetrical bimodal karyotypes in Epidendrum secundum. And we also have all this variation of chromosome numbers in pure populations or in hybrid zones. So it's too much, no? These are all from Brazil, just from Brazil. Imagine the rest. Santino is another species that we found to be probably a neohybrid too. You have this incongruence between nuclear and plastid data sets, but you know, incongruence between plastid and nuclear data set could be due to many other things. Misidentification, contamination, inaccurate pathology, also brain traction. For this, we just include several individuals from listed different places, and we found the same weird positions that are not expected. Branch attraction is not possible. We don't have long branches. So also horizontal gene transfer is not expected between two autotrophic species. So it means that we have incomplete lineage sorting or hybridization. So is it ancestral polymorphism? We test it using Pelser methodology that's simple and possible with Sanger sequencing that we have. So we have data from Fabio Pinheiro, that's also an author, and we were able to calculate effective population size. And we also date our trees separately, ITS and nuclear one. And we have generation time that we test 3, 2, 4, and 8 years. It's the time from seed to flower in this group. And we calculate estimated versus observed population size. And then we have results. So we are tending to believe that probably Epidendrum secundum and xanthinum are paleohybrids. I know that with genomics it will be much more strong, but we have cytogenetic data that more or less agree with the thing. This is the publication if you are curious to see something about it. And to finish, I think it doesn't matter if you are more splitter or if you are more lumper. In general, I think the thing on species delimitation is more about to think on taxonomy as science. So you need to test hypotheses, and each name is actually a hypothesis. So when you publish a species, actually you are publishing a name, and each name is a hypothesis. If it represents any species, that's the thing that you should test. Another thing that I would like to say is that it's for me, it's the most important thing. I have seen some presentations talking something the opposite from what I think. That I think taxonomy should inform conservation, not the opposite. For me, it's weird to think that we should keep some names just because it will be endangered. It's better to protect it. You are protecting just a name, you know, not a species. So with this, I finish my presentation. It's the people that helped me. I didn't do everything that I show you, so all these people are very important. And also other people that are not that important, but give me money to do that. So thank you.
Thank you so much. We have time for a couple of questions. Something? No?
Thanks so much, Edlley. It was awesome. Just wondering about plant variation in terms of the morphology of the stems, or maybe slight variations on the habitat, local habitat. I'm sure you have a lot of experience going to a field. What do you think about these, I don't know, big or small differences? Thank you.
That's something that I believe all biologists think about it. Why some species are very variable and other are not that variable, and how to deal about this. This variation represents different taxa or not. I think it's much more something to read in my ear than everything. I love my ear in general. It's just there are some for some reason, some populations change first morphology, but not there is no reproductive barriers. And in the opposite, some first reach reproductive barriers but are not lifting on morphology. So why it happens, I don't know. But I'm really more going to escape of morphology in general. Like if I have a pattern like the morphometric analysis, I would like just to check it with genetics, just see if you have the same pattern. If yes, good. If not, probably not. Just convergence or parallelism. That's how I think.
Another.
Ellie, I loved your talk because yeah, there's several things in common with stuff we're doing at Lancaster, and we worked on a review of some vanilla species, and found that they were extremely well. There was a hyperinflation of taxonomical names in the group, and we still need to do a lot of work in that sense, with a lot of taxa being described very locally, and of course being announced as extremely endangered. So I really like that aspect of your talk, how how the circumscription of taxa influences concerns and conservation concerns, and the reality of it is that we can't conserve every single species, right? We can't give the limited amount of funds that are available to all 30,000 species of orchids. So we really need to be careful with just assigning names for little patches of things. And so I really like that. Maybe we can talk about that a little bit.
Good,
It's a
good. Thank
comment,
you.
not a question. I'm sorry.
Maybe it's that.
Thank you very much, Edlley.